Laura Dormer, and I'm the editor of The Evidence Base. I'm pleased to be joined today by Qianyi Zhang from Flatiron Health. Welcome, Qianyi. To kick things off, please could you introduce yourself and tell us a little bit about your background?
Hi, Laura. Thank you for speaking with me today. My name is Qianyi Zhang, and I am a senior research scientist at Flatiron Health, where I have worked for over 5 years. Flatiron Health is a health tech company dedicated to improving cancer care and advancing research. We enhance oncology care by leveraging data, machine learning, AI, and real-world evidence to deliver personalized care and transform patient experiences into valuable insights. And beyond being an EHR company, we accelerate the development of new treatments and improve clinical trial processes and create new possibilities for cancer patients within a modern, connected oncology ecosystem. As a biostatistician by training, my role involves designing and implementing statistical analyses that leverage the Flatiron Health real-world data sets to understand the performance of real-world endpoints and inform their development to facilitate various fit-for-use real-world evidence research.
Now, you're here to talk to us today about the research that you presented at the recent ISPOR 2024 conference, and that was your research evaluation of real-world response rate in clinical trial-aligned cohorts of patients with lung, colon, and breast cancer using machine learning. So first of all, could you tell us what were the main objectives of your study?
Yeah, so in oncology clinical research, understanding how patients respond to treatment and the associated endpoints is very important. The rapid generation of this evidence using real-world data plays a critical role in informing the design of clinical trials and the development of new drugs. But within the context of clinical trial replication specifically, leveraging real-world data to understand response rate help us characterize the patient population of interest in the trial, which in turn informs decision-making and prioritizing drug development investments. And additionally, it allows us to assess drugs' effectiveness in real-world settings, providing a more comprehensive understanding of its impact. To achieve this, we developed a real-world response RWR approach using natural language processing NLP-based deep learning models. And these models, which were trained on expert human abstracted data, enable us to generate RWR in a scalable and expedited way. And as a result, we have introduced the term scaled RWR to describe this variable's ability to rapidly provide insights across large patient cohorts. And our study primarily focuses on evaluating the real-world response rate using this scaled RWR variable within clinical trial aligned cohorts. And these cohorts included patients, as you mentioned, diagnosed with lung, colorectal, and breast cancer across various therapy classes, and from the control arms of 4 clinical trials, with 2 that focused on chemotherapy and the other 2 that focused on oral targeted and hormonal therapies. And by analyzing these cohorts, we aim to enhance our understanding of the relationships between response endpoints in clinical trial and real-world data.
And could you describe the data sources that were used in the study and how they were selected?
Yeah. Firstly, we leveraged Flatiron Health's extensive de-identified EHR database, which encompasses structured and unstructured real-world data derived from EHR of patients treated at community oncology and academic cancer centers across the US. And additionally, we also utilized the published results from the clinical trials, including the summary statistics of the baseline characteristics as well as the endpoints results to help align the real-world cohort and to facilitate the comparison with the study results from the real-world cohorts.
How did you ensure that the generated real-world cohorts were aligned with the control arms of your selected trials?
We applied several steps to ensure that the real-world cohorts were aligned. First, we made sure that the patients in the real-world cohort received the same therapy as in the control arm of the trial and within the same trial enrollment period, creating a contemporaneous real-world cohort. And next, we implemented the trial inclusion and exclusion criteria wherever feasible on the real-world cohort. And finally, we also applied the inverse odds weighting method to further align the real-world cohort based on the baseline characteristics from those trials.
How was machine learning used to evaluate the real-world response rates?
Yeah, in this study, the machine learning approach, specifically an NLP-based deep learning model, was used, and it learned from expert human abstracted data to extract response assessments from a vast amount of unstructured EHR data, including radiology reports and oncologist clinic visit notes. And then we use these machine learning extracted responses to calculate the real-world response rate.
And what were the key findings of your study?
Yeah, our study revealed several important findings. First, in the 2 chemotherapy-focused trials, the overall response rates closely matched the real-world response rates. However, in the 2 trials that focusing on oral targeted and hormonal therapies, discrepancies were observed. And we hypothesized that the differences stem from the challenges in replicating some trial criteria in the real-world setting. For example, the variations in the biomarker status determination impacted patient selection and subsequently real-world response rate. And furthermore, the adherence to oral therapy, which is crucial for treatment effectiveness, proved to be more challenging in the real-world settings compared to clinical trials. So this difficulty potentially influenced the real response rate, particularly when follow-up assessments were needed to confirm the response. And we also recognized the nuances involved in the comparison and applied multiple approaches to address these complexities. So one of the ways was to calculate the real-world response rate by including or excluding patients without any response assessment, which gave us a range of results with the true value likely somewhere in between to account for the potential missingness of assessment in real-world settings. And additionally, we look at real-world response rate with and without confirmation, meaning that requiring a subsequent response time point after the initial response. And similar to how that concept is captured in some trials. For instance, in the oral hormonal therapy, real-world response rate closely aligned with overall response rate without confirmation, but it dropped when confirmation was required, likely driven by different cadence of imaging in the real world versus trial settings. And overall, these findings showed how important it is to consider the nuances in real-world response rate calculations when comparing the results between real-world and clinical trials. And they also highlight the need to carefully evaluate the inclusion and exclusion criteria, as this can vary based on the patient population. And lastly, our study also should suggest that the patterns of real-world and trial outcomes might differ across different therapy classes.
So what future research will be needed to take this work forward?
Moving forward, there were several avenues we could explore. First of all, we hope to broaden the scope of this study by expanding our analysis to include a wider range of cancer types and therapy classes. This will provide a more comprehensive understanding of how using endpoints impact outcomes in diverse settings and identify similar or notable trends between real-world data and clinical trials. While our current analyses have relied on standard Flatiron Health EHR datasets and the published trial results, we also recognize the potential benefits of capturing additional data elements in the real world via abstraction or ML extraction, also the benefit of gaining access to patient-level trial data. So because both of these could enhance the robustness of our matching techniques and lead to more reliable results. And lastly, our team is also exploring the use of ML AI techniques to develop and scale other variables and endpoints in the real world. For example, real-world adverse events. By generating these high-quality variables, we aim to facilitate research using real evidence with greater precision and depth.
So we've spoken today about this specific piece of research, but do you have any other prior publications in the same field?
Yeah, we have several prior publications in this field. And before developing the scaled RWR, we developed an approach called detailed RWR, which relies on expert human abstraction. And we have been publishing our findings to illustrate the journey and the progress with real-world endpoints. To name a few, one of the earlier publications focuses on the developing and assessing the methodology of detailed RWR. It also explored how the overall response rate compared to the real-world response rate in clinical trials for non-small cell lung cancer. And this work was published in Advances in Therapy in 2021. And building on that, we conducted a clinical trial replication and endpoint comparison with the access to patient-level trial data for a specific trial. And this study was published in JAMA Network Open earlier this year. And more recently, we built on the detailed RWR and developed the scale RWR variable, which offers greater scalability. So in this publication that we're talking about, we were able to assess real-world response rates in trial-aligned cohorts across a broader range of diseases and therapy classes. And these publications all highlight our ongoing effort to bridge the gap between real-world data and clinical trial findings for oncology.
Thank you. And why is it that you think that this type of research is important?
Yeah, there are numerous important use cases for real-world endpoints and broader real-world evidence research, as it helps us to understand the patterns of care and patient outcomes outside the clinical trial settings, including gaining insights into treatment selection and sequencing, as well as identifying and characterizing population with unmet medical needs. And more specifically, this type of research is crucial for informing internal decision-making and investment prioritization for biopharma companies. For example, if we found a cohort of patients with poor response outcomes in a later line setting, it can inform potential areas for investment in novel therapy development. And also, it supports patient and provider decision-making at the point of care, such as offering critical information for more effective treatment choices. And also, real-world evidence research is vital for understanding the patient's journey from diagnosis through treatment and beyond. And lastly, the endpoints, for example, the real-world response, are very essential for conducting comparative effectiveness analysis and characterizing the clinical effectiveness of targeted drugs in the real-world settings. So by leveraging the ML and AI techniques, we anticipate accelerating the value this type of research could bring to us for drug development, clinical care, and ultimately enhancing patients' experiences.
Thank you. Great. Well, thank you very much for talking to us today, Qianyi. It's been really interesting to hear more about this work and some of the potential future efforts as well. So I'd just like to close by thanking our viewers for joining us on the Evidence Base today and thanking you again for speaking to us. Thank you.
Yeah, my pleasure. Thank you for having me today.